I just wanted to start out by saying thank you so much for coming. I really appreciate the turnout, it being Mother's Day, so happy Mother's Day to all the mothers in the room. I'll get started. So I want to start out by just head-on addressing a potential misconception about the title of the program that I'm talking about. So OPIHI is a culturally important limpet here in Hawaii, while OPIHI the acronym stands for Our Project in Hawaii's Intertidal. And the acronym was, of course, chosen in part based on where the OPIHI limpet lives and its importance. But confusingly, in the program OPIHI, we don't actually really see the limpet OPIHI. And that is because, as I'll explain a little bit later, we've mostly worked on Oahu, where OPIHI are very rare. And we've also mostly worked with students and taken them to safe intertidal areas, where OPIHI are really rare. But I'll talk more about our expansion of the program, and maybe we'll see some of these more in the future on other islands. So because this is called a citizen science story, I want to tell you a little bit about my story and where I'm coming from. I'm really passionate about this program, so hopefully this will explain my passion a little bit. And this way, hopefully you'll also feel more free interrupting me if you have any questions. So while I was doing my master's research here at UH on urchins on patch reefs, I actually spent more of my time in the intertidal. And this was for two reasons. One, I found out I was really allergic to many things in the water. So I have my representative Portuguese man-o-war out there where so it might hurt you a lot, but I go into anaphylactic shock. So I was becoming a bit of a liability as a budding marine scientist who had trouble getting into the water. A bit of a career conflict. And I became involved in a National Science Foundation program where they paired budding scientists like myself in grad school with teachers. And the idea was that the teachers would teach the scientists how to teach about science. And then the scientists would teach the teachers about the process of science. And it was just this amazing program that really came to embody both of my loves, which are science and communicating science and that education aspect. And it also allowed me to go into the marine environment without actually really getting wet. And so as you can see, in this picture, when I started doing intertidal work, I still wear high boots and I wear gloves. It's not fun to go to the hospital in the middle of a field trip. And nowadays I actually wear pants and long sleeves too, so I really look like this interesting intertidal ninja. But it's a way for me and other people who are not comfortable swimming, and that especially is for people and students in the Department of Education. So it's a marine experience that is very safe. Yes?
Can you just explain intertidal?
Sure, it's actually the next slide. So you're segueing very, very well. Oh, it's not the next slide. It's the slide after this. But I'm going to start out with what is the intertidal. Then why is the intertidal important? What is citizen science? And then segueing into our project in Hawaii's intertidal 10 years ago, and I'm going to show you how the project started and what we've found. And then it's being revitalized and what our hopes are during this revitalization. And I'm going to end with, we need you. So a little of a pep. So the intertidal is a unique marine environment that lies at the interface of land and sea. So it's covered with water during high tide, and it's exposed to air during low tide. And tides are predictable daily rises and fall of large surface waters like the ocean and large lakes. And they're predictable because They're based on mostly the celestial movements of the Earth, Moon, and Sun, and they're actually waves. So high tide is when the tidal crest, that wave crest, arrives on shore at a particular location and it raises the sea level. And low tides are during the troughs of the wave, where it lowers the sea level. And the difference between high and low tides is called the tidal range. So the tidal range and thus the size of the intertidal can vary really dramatically based on location. So it's based on the movement of the moon and the earth and the sun, but also a lot of other factors like the tilt of the earth, the location in orbit of where we are around the sun, where the moon is around earth, and also geographic factors like coastline and seafloor features, as well as wave action. So for example, the difference between high and low tides in the Bay of Fundy in Canada can be as large as 16 meters, which is the height of a 3-story house. One of the reasons is because the bay is funnel shaped, so it actually kind of funnels the water up. At the back of the bay, it gets very, very different in terms of tidal highs and lows. However, other areas like the Mediterranean Sea only maybe have an intertidal of a few centimeters different, just because of its location, and all the water has to move through that Strait of Gibraltar. So the tidal difference is not very big. In California, where there's been a lot of intertidal work, the tidal range is about 7 feet or about over 2 meters. So here in Hawaii, our tidal range is about 1 meter or 3 feet. It's a pretty small tidal range, but you can see the difference between high and low tide. This is on Mealy Point on Oahu. So it's not super dramatic, but based on the coastline, it can definitely, you can tell in pictures, the difference between high and low tide. So in areas with really large tidal ranges, like in California, and large tidal ranges tend to be more in northern latitudes, there may be clear biological zones designating high intertidal, low intertidal, and mid-tide zones. But in Hawaii, especially on really flat benches like this one, these zones can be blurred just because of our small tidal range. But they're more readily visible on steep rock faces like here, where the colored bands in these zones actually correspond to characterizations by different species. So within each zone are creatures that have adapted to thrive in that unique environment based on air and water exposure. So the intertidal has some pretty unique organisms, and most of the animals and plants that live in the intertidal are marine in origin, which means that they need water to breathe. And so the intertidal can actually be viewed as a bit of a stressful environment for them. They have to deal with waves that could knock them loose. They have to deal with being exposed to the air for sometimes large periods of time. They have to deal, during that time, they have to deal with the stress of not drying up, temperature changes, particularly on very sunny days here. During very low tides, this is associated also with changes in salinity. If you're stuck in a tide pool kind of high in the intertidal, you could get more saline as things evaporate. And they're also exposed to both land and sea predators. Thus, the organisms in the intertidal have a lot of unique adaptations for this challenging set of living conditions. So many adaptations are to prevent water loss. So for example, this is an intertidal snail, and this is the operculum. And it can actually sort of seal itself up inside the shell and prevent water loss that way. There's also some behaviors that can prevent water loss. So these are littorines, also called pipipi, or what are these? Neurite. No, not neurites, but neurites do this too. And they actually, you can see them clumped. There is like a shadow here, and they've actually clumped more on the dark side of the rock. So they're trying to avoid water loss and moving around this rock based on sun exposure. Here, this is an anemone, and of course they're soft-bodied, so they're really susceptible to drying out, and it'll actually pull in its tentacles and kind of shrivel up a little bit to prevent water loss. Other adaptations are to prevent waves from knocking them loose. So helmet urchins up here don't actually have spines on the top of them. They're sort of evolved into plates, and they can really suction down and move those plates together and suction down in the intertidal, so they don't get knocked off, but they also don't lose water. Other urchins, and you can't really see them because they're in burrows here, so they're sort of had this adaptation where they live in these burrows. Which helps you when you're walking around so you don't get stabbed by them, but also prevents waves from knocking them off. And algae, of course, they can't go in burrows because they need the sun, but a lot of them are flexible, so they'll have a hold fast and then be very flexible and be able to bend with the waves. And others of them, like this is actually algae right here, this pink rock, it's called crustose coralline algae, and it's almost like a paint on, so obviously that's a really good way to prevent being knocked off by waves. And they're also very camouflaged for predators. So there's actually a fish in here. This is a sea cucumber and a crab. So why should we care about the intertidal? For one, it's an important part of Hawaiian cultural practices. So Hawaiians harvest limu, also called native algae, for food as well as medicine and religious and ceremonial purposes. So for example, limu kohu, which is here, is really fluffy. The scientific name is Asparagopsis taxiformis. And you can rinse it and then soak it in fresh water, salt it, pound it, roll it into balls, and add it to things like poke or stewed beef. This is a very prized and hard to find sometimes on Oahu. It's a type of Grasslaria cornifolia, and it's called limu, and I'm going to masticate this a little bit, Manuea. And it's also, this is really common for poke also. There's also something like sargassum, which is up here, limu kala, which is used a lot for ceremonial purposes. Cowries for jewelry and for scraping, as well as octopus lures. You can eat things like urchins and sea cucumbers. The intertidal is also really important ecologically. It can serve as a nursery for fish that live in the subtidal when they're adults. And it has a lot of native and endemic species. So all of the limu, except for one, it only occurs here in Hawaii, so pretty special. So because it's at this interface of land and sea, it bears the brunt of the impact of both terrestrial or land and marine impacts. So it's really susceptible to human threats. Because it's at the base of the watershed, it's affected by everything that happens upstream. So development, urbanization, and eutrophication. So during urbanization, when you pave different areas, freshwater runoff, instead of being soaked into the dirt, can run off and make water really cloudy as it carries dirt with us. Muddy water can block and kill coral. In addition to things like fertilizer runoff from like golf courses types of things, Hawaii's had a really long and sort of well-known problem with sewage in coastal waters, including direct disposal into estuaries, sewage spills, injection wells, and leakage from cesspools. occurs not just on Maui and on Oahu as well. And this submarine groundwater discharge is a major vector for delivering nutrient-polluted groundwater into coastal ecosystems. And tropical seaweeds, unfortunately, respond very well to this, and they can increase their growth and in response to these increased in nutrients. For instance, this leafy green algae right here, it grows really well in areas with a lot of nutrients from fertilizer runoff, and it can also survive in areas with a lot of fresh water. So if it's increasing over time in an area, that can be indicative of enhanced nutrients or enhanced runoff. And because the intertidal is so accessible, it's often affected by over-harvesting as well. So for example, the Blackfoot OPIHI are essentially extinct on Oahu due to over-harvesting, and they're really severely depleted on other main Hawaiian islands. Conservation efforts are constrained for this organism because it has a very limited larval dispersal. So when it releases eggs and sperm into the water, they don't travel very far. They sort of stay on the island. And so it's this really strong population structure. The Maui population is different than the Oahu population, is different than the Kauai population. When an area goes locally extinct, it's very hard to bring them back. Trampling can also be a problem. If you go to if you go to the intertidal at all, you can see a lot of school groups there sometimes during spring low tides. And I wanted to read the caption for this one. I pulled this offline. And the person writing the article says, "The kids searched every corner and crevice looking for sea life they could see and touch. And they all had nets." It's like 50 kids. Like, oh. There's like a good way to explore the marine environment. And then there's the, I don't know, like rape and pillage way or something like that. And certainly I'm advocating that students go into the intertidal, but I think definitely to be aware of the sustainability factor of going to these environments. The intertidal is often the first area of appearance for alien species. Alien species are species that have been introduced to Hawaii, often in this environment on boat hulls. And although some of them seem to have very little impact, others can actually become invasive. And there's quite a number of invasive species in the Hawaiian intertidal. So this is an invasive species called Hypnea muciformis. It's got these very characteristic hooks on it. It thrives in nutrient waters as well. And endangered sea turtles that consume this species sequester excess nitrogen, which promotes a virus that can lead to debilitating tumors and death. If you've seen the sea turtles with tumors, this is one hypothesis for how that came to be. It's also very expensive to deal with invasive algae blooms. Over 10 years ago, Maui was spending $20 million a year on dealing with these, and I would imagine it's even more now. There's also other, not just invasive algae, this is an invasive barnacle. It wasn't in Hawaii before 1995, or at least wasn't noticed, and now it's sort of invading some of these rocky intertidal zones. Also, climate change. So climate change is likely to cause complex chemical, like ocean acidification, and physical. So it's hypothesized that current patterns will change in coastal marine ecosystems. And the degree to which intertidal species will respond to these changes, like sea level rise and air and sea temperatures, is really not clear. On one hand, the very nature of this zone defines its resilience and its potential to adapt, and it's had to respond to land and sea fluctuations for its entire existence. And so this position is supported by some long-term monitoring studies that have found little changes in species. But other studies have found really dramatic changes. For instance, there was a study in California, and over 60 years, they noticed basically a northern migration of species, and they hypothesized that was in response to climate change. But in island ecosystems, intertidal organisms can't move. They're kind of stuck here. So they either must respond to climate change or adapt or go extinct. So it's really vital to monitor this ecosystem, especially as it's so narrow. And this is just Chip Fletcher. He's at UH, and he's just showing the approximate sea level at the Hawai Canal in the next 100 years. So just a summary about our background information. The intertidal zone in Hawaii is small, but it's home to species with cultural, economic, and ecological importance. Due to its location, it's impacted by both land and sea issues. But I really think it's a model habitat for measuring ecological change. So just change in the number and amount of species over time. But it's actually been really poorly studied. So in general, tropical intertidal zones are not as well known as temperate or more northern intertidal zones. And that's partially just because of size, it's smaller often in the tropics. And we have some really great coral reefs just beyond our intertidal zone. The water is not cold like in California, so more likely to want to jump in and do research here. So, and because of that, it's really an inconspicuous zone. It's not very well known and it's not very well studied. Another thing that makes it very difficult to sample or study is due to seasonal wave activity. So if you just think, when are the waves really high on the North Shore? When are they high when is there a south swell? It varies with the seasons. And there's actually a very small low tide window in a very small area. So this graph shows the number of days each month with negative low tides. It's a little old in 2007, but they haven't changed very much. So the intertidal is only exposed for a few hours every day with negative low tides. So these are the negative. So basically the way to look at this graph is the best times to go out in the intertidal are when the days are red. So you can see that they are in spring, and there's just not very many of them. So surveying this type of environment requires a large number of researchers to work together during these very limited time periods. So the type of long-term monitoring needed to detect change and provide insight as to the causes of that change is really difficult to implement. So what we really need is a cadre of trained citizen scientists who can collect data over a wide geographic area in a short time period on the best low tide days, something that would be too costly, too time consuming, and too labor intensive for researchers. Citizen science is a partnership where the public and professional scientists come together for mutual goals. So the public is trained in how to collect scientific data and has the opportunity to engage in authentic scientific research. Like, I literally do not know what we will find. It's not hard to beat around the bush with that one. Scientists are also able to collect a large amount of data, often in a relatively short period of time, and address questions that would otherwise just be logistically unfeasible to do. Citizens involved in citizen science, there's a lot of benefits. So this is why you should participate. I'm going to read some of them. Increasing awareness and understanding of environmental issues, developing an active interest in nature, increasing connectiveness and responsibility to local marine environments, an ability to communicate science and that experience to others. And basically, we're creating a more scientifically literate society. You guys have the opportunity to be on the forefront of that. It's not a new concept. For instance, these birders are involved in the Audubon Christmas bird count, which has actually been going on since 1900. And it's collected just a vast amount of data. And people are really utilizing that data set now. There's also things you also may have heard of on the mainland. There's a very popular monarch butterfly project. Here in Hawaii, we have Project Budburst, where you can see when things are blooming and mark them and see if that's changing with climate change when things are blooming. There's also what are kind of called bio-blitzes. There's actually one happening in Volcanoes National Park, I think this week, where a lot of people go out and try to ID everything in an area. And the Intertidal is a really great place to do citizen science because even though it's very accessible, which can be a bad thing in terms of over-harvesting and trampling, it also makes it an ideal location to take students. So as I mentioned before, it's Department of Education approved. Students don't get wet past their knees, and there's no need for lifeguards, which is a big hang up when taking students into the marine environment. So what were the goals of this project? At the start of the project, the goals were actually to just describe this ecosystem, what is there. So making a list of the invertebrates, the fish, the algae, how invaded was the intertidal, basic concepts like were there endemic species, what % were the endemic species, and those are species that are just in Hawaii. Are there differences between sites? What's the most common species? Things that you'd think we'd already know, but we didn't. The educational goals were to generate enthusiasm for and confidence in doing science, practice really rigorous science, create awareness of the ecosystem, and increase understanding of basic science concepts. So students planned and actually carried out the investigations themselves, they ID'd the organisms, and they analyzed the data. And actually this first cohort presented their results at scientific conferences. So, and because this was the first description of the intertidal, it would establish a baseline from which changes over time could be measured. So this project started over 10 years ago in 2003, and it was very much like a bio-blitz. So students from 2 marine biology classes at one school, they were in 9th grade, were split up into taxonomic teams, and they ID'd with the help of scientists, including those at Bishop, everything that they found down to species level, which is like a very exact scientific way of categorizing an organism. 9 sites were sampled on Oahu, and over 500 species were identified. And the number actually didn't flatten out. Every time they went to a site, they found more and more species. So there's still more that hasn't been classified in this environment. This is the number of species is actually very similar to temperate regions like California, which is unexpected. It's just that California has more larger organisms, so the size is sort of more dramatic than we have here. Also, we have a very high number of invasive and endemic species. You can see that we looked at sites on all sides of the island, but analyses actually showed no obvious patterns among the sites with things like wave action. Educationally, this graph is from a case study of the students, and they had significant improvements in concept and skills knowledge. The blue is post, and the white is pre, on concepts related to intertidal knowledge and ecology. So starting in 2004, to expand the program, now that we knew the species that were there, the protocol was more standardized to collect abundance data, so things like how much is there. So we use standard ecological monitoring tools like transects, which are essentially measuring tapes, and quadrats, which are PVC in a square. But to call them scientifically, we call them quadrats. And from 2004 to 2007, students from 8 middle and high schools from across the Hawaiian Islands monitored 12 intertidal sites, some of them up to 10 times each, grades 6 to 12. And this was the first description of community level patterns in the intertidal. We also standardized classroom preparation at this time because when you increase effort, not every student group can be partnered with a Bishop scientist. So they had standardized lessons that they went through where they learned what sampling is, or how do you little aspects, little areas of the environment, how does that represent the entire area? General ecology, so like things like predation, competition, and why monitoring this area is really important. They also learned how to identify invertebrates and algae. They practiced using identification cards and books, and practiced the techniques, so going outside and practicing things. In the field, they laid out tapes, so perpendicular to the shore, and there would be multiple tapes. And they would place these quadrats along the tapes at set intervals. And then you would look to see what was underneath. So you can barely see it in this one, but in each quadrat, there's fishing line that creates a grid of 25 points in the quadrat. And under each point, students would identify what was there. So this way, we're looking at a small piece of the intertidal to try to characterize the whole thing. They worked in groups, often with a science assistant, to help the students stay on track, take quality data, sort of point out like, oh, you know, maybe let's look in the book for this one. It doesn't seem to be on the card. These science assistants included undergraduates, graduate students, professors, community members, and parents. So again, we wanted to look for patterns across sites, because the first time we looked at that with just lists of species. The first time was literally just what is there, what species are there. But this time we had some more information in terms of abundance, how much is there. So for instance, we wanted to examine bench sites. So these look like benches. They're very flat, hard, intertidal areas, and we wanted to see if they were similar and if they would sort of clump together in terms of the species at these locations. Do they have kind of the same types of species? So this is a kind of overwhelming graph. But it's basically each of these points indicates an average by year of species. So we're taking all of the species that we found at a site and we're sort of narrowing that down to one point. And these are, unfortunately, acronyms. But I wanted to show this to basically show you guys that there's really no clear clustering of the sites. So those bench sites on this slide kind of cluster here. These are bench sites as well. So those really broad categories that we were looking at in terms of, was it a bench site or was it boulders, which are kind of like rocks? Was it on the north shore versus the south shore? Those really broad characterizations weren't really teasing out patterns. There's some clumping, maybe some like calm nutrient type of sites, some more invasive, but nothing very clear and nothing significant. However, we were able to show that there is zonation in Hawaii's intertidal. And zonation is very obvious at areas with very large tidal ranges. It's not very obvious here, but at some sites you can see it. So Diamond Head, which is just down the road, and Barber's Point have these nice zonation patterns where some algae are more abundant in the lower intertidal and some algae are more abundant, in the lower, and some are more abundant in the more exposed areas. So basically what we knew in 2007, Hawaii has a really rich intertidal assemblage. There's evidence that sites group by environmental factors. There are patterns, but they're not consistent, and nothing was really being teased out. I do just want to have a fairly quick mention of the validity of student data. So one of the primary challenges when conducting a citizen science project is to address the scientific community's concerns about the data. And when questions arise, the data doesn't seem to be utilized or very underutilized, which is detrimental to both the educational and scientific goals. So very few studies have rigorously analyzed the validity of the data, but we did. We directly compared scientists and students in 2007. So there were 2 teams of experts, and these 2 teams, as well as students, all collected data along the same transect. And you can see the students are here, the researchers, one of the teams is here. It was a logistical nightmare, but we were able to answer the question, is student-generated data comparable to researcher-generated data? And the short answer to that is yes. This scatter plot is of diversity values. So again, you're taking everything that we know about a site in terms of the number of species and how abundant they are and putting it into one point. And here, there's different sites. The researchers are in black and red, those 2 researcher groups, and the students are in green. So you can see at some sites, they all kind of clump together. They're basically all collecting almost the same data. At other sites, they're kind of more different. But the students are very close to the researchers. So we were able to say statistically that while the researchers' values actually varied more from each other than the average between the 2 of them and students, which is pretty amazing, based on a very short period in the classroom of prepping for this program. Students did miss some things. So these are organisms that were found by the researchers that were not found by the students. And don't pay attention so much to the scientific names, more the pictures. These are little worms. You really have to know what you're looking at in order to identify them. This algae is small and fairly flat and very brown. It doesn't really stand out. And crustose coral and algae is sort of like that pink paint, and unless you know it's an organism, it's hard to write it down on your data sheet. So students, they do miss some small cryptic and low abundance species. So, but sometimes students recorded data that the researchers did not. And interestingly, this mostly occurred, so all of these are above 1%, in invasive species. And this was all at one site. And what we figured out was this teacher really wanted her students to understand that invasive species in the intertidal were a bad thing. And she wanted them to be able to recognize them and see how they were affecting the environment. So the students got really overzealous and identified these when they were actually not invasive. So they're actually, these guys are all kind of close in terms of how they look. And so I think there was some confusion between them and also with native species that can also look similar. So there's a little bit of overestimating of invasives. So basically this assessment demonstrated that middle and high school students, this is starting at basically 11 years old, are very similar to professional researchers when you're looking at more common species. So they're good at identifying large common species, but maybe less good with new introductions or rare things. So we've learned from this to really underscore the importance of proper identification with students, emphasize negative consequences, and provide explicit instruction in how to ID things. Not doing very well, but now we're looking at revitalizing the project. So it's been over 10 years, and I'm really excited about the opportunity to do this. Those surveys from 2004 to 7 are actually going to serve as the baseline for our proposed assessment. And we're going to see how things have changed over the last 10 years, which is, you don't get it, monitoring studies, they're really hard to find money for because you're not necessarily testing these really amazing questions, but change is very important. So citizen science is a really great way to do that. We will provide recommendations of ongoing and predicted effects of coastal development and climate change, mentor the next generation of community ecologists, and we're going to update the protocol a little bit. 10 years ago, water quality monitoring was more expensive than it is right now, so we're going to add a bunch of water quality things. So that includes things like temperature, pH, salinity, and nutrients, which will be added to the protocol, because these things that we already looked at, slope, substrate, in terms of if it was basalt or black, and limestone or coral generated, and wave action weren't really teasing things out. So there's a more fine scale level that we need to go to see differences between the sites. We're also going to construct a public database, which shall be available to everyone, including those in the project, so they can monitor, they can look at sites that they haven't necessarily go to, but people who are doing similar things have. We're going to update our monitoring cards. Some of the species names have changed over the last 10 years. We're also looking into maybe an app or an intertidal guide. And we're going to strategically join forces with other organizations. So there is something called, even more confusingly, the OPIHI Monitoring Partnership. It's run by the Nature Conservancy, and they do go to those crazy vertical areas, mostly on other islands, to look at the OPIHI population. So we're actually going to join with them and compare protocols, and they're going to do what we do and we're going to do what they do to see if we find different things, and also just leveraging resources and sharing people.
What does the OPIHI tell you whether it's prevalent or not? Why is it important?
It's important as a cultural resource, and there are groups that are recognizing that they're just not abundant anymore. And so a recognition that we've over-harvested this, and if we set up rest areas, are the organisms going to come back? How large do they need to be? We don't even know. I mean, we're just starting to even recognize how prevalent these are. Until recently, I think people assumed there was sort of more on Oahu than there actually are. Like surveys have gone on Oahu. There's 3 species of OPIHI, and one of them is the one that's in the highest intertidal. It's just not here anymore, and it's not going to come back. So what can we do on other islands to sort of protect this species? They have a Facebook page. It's very exciting if you want to see scientists jump off cliffs. And they are also a citizen science monitoring project as well, but more active on Maui. So if you're a teacher, anyone here a teacher? We need you. Okay, tell your teacher friends. I'll just skip this 2 slides. But we also need scientists and science enthusiasts. So I really think, your ability to help with the students and would be just a great resource. Basically, it's pretty easy. You help ID organisms, but you don't have to be a scientist. We have ID cards the same as the students. It's more of when the students seem to jump to an identification and you're like, let's look at the ID card and maybe we want to revisit that. You would help small groups of students of about 3 to 4 follow the protocols as you go along a transect. There's no prior knowledge required. It's really learning about your area. We're going to be going to sites across Oahu and expanding to the other islands as well. I think it's pretty fun. It's only about 2 to 3 hours because the low tide window is so small. They're generally during the day, school hours, of course, but let me know if you're interested. And a small number may actually be able to attend the workshops we're going to give to teachers as well if you're interested. I have a sign-up sheet here if you'd like to be put on our mailing list. So with that, thank you so much, and let me know if you have any questions.
Questions. What is a sand scour?
Oh, so the bench sites that I showed you in that one diagram don't have a lot of sand on them. So they kind of go from dirt, and then there's like a bench of, it's like basalt or limestone, and there's not a lot of sand on them. Whereas some of them, like Makapu'u, has both sand and sort of a rocky area. And so the sand can scour that rocky area. And then there's, of course, sandy sites, and we don't go to the sandy sites. We just stick to the rocky intertidal sites.
You talked about using this data to establish safe zones that would protect species such as OPIHI from harvest. Roughly, what's the time frame between gathering and publishing all this data and then enacting legislation?
Oh, that's a really good question. So in 2003, there were, I don't want to say there was no intertidal work done, but there was very little. Now there are 2 rest areas on Maui for OPIHI. Now that's not because of this project, but because it's the interest generated from this project. I think it really sparked a little bit of a resurgence in intertidal work. The person that's sort of running that CRISPR was actually a science assistant in the very early stages of the project. On Oahu, we have had people like the Nature Conservancy just interested in what are the hot spots of intertidal diversity on Oahu, but it's actually not a question we can really answer yet. We're like, what kind of area do you want to protect? And yeah, but in terms of the time frame between collecting and publishing, the surveys ended in 2007 and the paper wasn't published until 2012. So yes, there's a delay, which is why we want to get the database up. So the database, teachers and students and the public could upload data. We'll have some filtering procedures, so we know that it's valid, and then anyone can start looking at it very quickly.
Is there a website that we can use to see when the tide is?
Yeah, NOAA.
That's the best.
Yeah, NOAA Tides and Currents. And then choose a location near you. They're actually pretty good. Now's the time to go. Spring and summer.
Do you tell your teams to just be careful where they step? I was thinking, gosh, you're going to be stepping on things, but of course you have to be out there.
Yeah, so that's
you have to step on the rocks. You just have to kind of watch out for the critter, right?
It's definitely something that we think about, and we've started doing more of a sort of a reflection before and afterwards in terms of when you go to a site, look at it now, you're going to impact this site. The good thing a little bit about the intertidal is that it is sort of hardy in terms of some organisms, not all of course, but some of them. But yeah, we're totally impacting the environment, and trampling does affect it. We try not to take large groups out, 20, 25 students at most, and try to stay away from and we don't have nets, we don't try to collect things like that. Because of the type of monitoring we do, we mostly see algae because there's no fish that's going to hang out in a quadrat, for better or worse. We don't capture them a whole lot. But we have done separate fish surveys with researchers. I think it is important to go, though, because teachers do shy away from marine field trips because of the lifeguard component. And if you're not going into the marine environment with students, you're not exposing them to that environment, which is so important here in Hawaii. And so I think balancing that impact and educational aspect. We also, we try not to go to, the places that we go to tend to have bathrooms fairly closely, so they're the more impacted area. We have another grant that will be, we're hoping to get that will be more just undergrads, and it'll be a smaller group. We can go to some more unsafe zones because they have bigger bladders.
With regards to the nets thing, is it more because there's a fear of catching and not releasing, or just disturbing the habitat and the quality of the data?
Oh, I'm not against nets. I think I'm against 50 students having nets and indiscriminately going out, because you cannot monitor 50 students. And the ones that tend to go out with these sort of large groups are your younger students. So when you see intertidal groups, it's more elementary school. And I'm not saying don't take elementary school students out, but they
require sufficient supervision.
Yes. And so I think it's just being very aware of how you're going to look at organisms. Being aware that when you take something out in a look box and you put it on shore so everyone can see it, if it's a hot day, that water is going to get hot pretty quickly. what kind of oxygen needs might this organism have? There's also, people tend to take some of these things back to the classroom, and some things just don't do well in tanks either. Like sea cucumbers just are really hard for aquarium
saltwater
people.
tanks in general are difficult to maintain.
Yeah. So there are some like urchins and stuff like that. So I'm not saying no tanks either. I'm just saying on our website we do have a list of things that do better. Like hermit crabs, pretty much impossible to kill in a tank. Good to have in a classroom. Eat a lot of cool stuff. Interesting behaviors. I mean, I don't want to take away that behavior aspect either, I think. And I think I think in general, like, when people are realizing that stuff more now, we just want it to filter down.
Last week, the presenter talked about coastal flats. Is that anywhere in this area right here?
Coastal flat.
Yeah. For
He talks about OEO,
OEO.
the bonefish.
Is that in the same area here? Or is that
It would be just,
further out?
it would just be just beyond that before you hit the coral. So it actually would be like, have your intertidal zone, and then you have your kind of sandy flat, and then you have your corals. Yeah. That's a good question though. Any other questions? All right, thank you, Joanna.
Thanks so much for coming, and please sign up.